According to Tracy Staditer, scientists have been using CRISPR technology to, quote, snip out and replace DNA to develop treatments for cancer, HIV, chronic pain, muscular dystrophy, and Huntington's disease, end quote. CRISPR technology is a powerful tool that will eventually dominate in the near future and help individuals get rid of undesirable traits and diseases. I believe that CRISPR technology should be accepted in society today, and my stance on this is based on excessive research and factual evidence that I have gathered. And today I will be informing you of this factual evidence in an unbiased manner. Clustered regularly interspaces short palindromic repeat, also known as CRISPR, is a technological advancement that can be used to eradicate life-altering genetic diseases as well as cause religious and ethical concerns while at the same time benefiting food industries and agriculture. CRISPR technology enables scientists to modify DNA sequencing in order to prevent undesirable genetic mutations. According to the article Rewriting the Code of Life, Jennifer Duwadna expressed, quote, CRISPR has been used to remove or correct the mutation in human cells that are responsible for cystic fibrosis, sickle cell anemia, blindness, combined immunodeficiency, among many other illnesses, end quote. Imagine if there was something that you can do for an individual in order to prevent them from having to suffer from a life-altering genetic disease. With an all-time increasing pace to discover cures and treatments, CRISPR technology is a promising innovation, especially for organ transplants. In the online article, Public Health Application of CRISPR, authors Vivian S. Vigligotti and Isabel Martinez statistically concluded that in the U.S., 1 in 10 infants less than 4 years of age died while on the liver transplant list, end quote. This is tremendously heartbreaking. However, with CRISPR technology engineering, they are able to reduce the shortage of organ availability for children and other individuals in need of it. And this is done through xenotransplantation. Now, xenotransplantation is an organ transplant between two different species. An example of a xenotransplant recipient would be a pig. Now, pig organs are suitable for humans. However, the porcine endogenous retrovirus, or PERVs, found in pigs pose an extreme risk for a successful organ transplant in humans. CRISPR technology, on the other hand, is able to remove this genome from pigs and make their organ a perfect match for individuals, therefore helping children in need of an organ. Although CRISPR technology can benefit others from its genetic alteration, it is also a controversial topic among many individuals. CRISPR technology surpasses the norms of society by advertising the modification of undesirable traits, which uprises religious and ethical concerns. In the article, Should CRISPR Scientists Play God?, Ted Peters suggests that, quote, laboratory scientists play God when they alter life and influence the future evolution of the human species, end quote. Controversies over CRISPR technology hinders the moral recognition of religion and how intervening with nature goes against God's distinctive plan and image for creation. Scientists are playing God toward deciding which hereditary factors individuals will have, therefore shaping an individual's personality. Religious activists uproar the fact that self-destruction is going to be heaped upon society for trying to intervene with God's image and plans. Another controversy over CRISPR technology is the ethical concerns for future generations regarding embryonic gene editing. In the article A Brave New World, Claire Wilson brings forth the concern how, quote, artificially induced mutations and their unknown effects on health may be passed on to the next generation and ultimately become a part of the human gene pool, end quote. Altering and editing genes in an embryo is a permanent effect. And the greatest ethical concern is the future impact these edited genes will have when these offsprings reproduce edited genes. Despite religious and ethical concerns, CRISPR technology prevails with benefits, especially regarding gene editing in plants. CRISPR technology is not just used for human gene alteration, but it is also used in food industries and agriculture. According to Ismail et al., the emergence of CRISPR genetic engineering, quote, significantly benefited the food industry by improving essential agronomic traits such as herbicide tolerance, insect resistance, grain yield, plant height, and weight, end quote. Improvements in food industry will amplify the quality and quantity of product production. With the increased demand for food, CRISPR technology utilization aids in meeting consumer consumption and growth demand. In conclusion, CRISPR technology can re-engineer disease-causing genomes, causing ethical and religious concerns, yet prevail in sustaining food and agricultural demand. Technology is constantly advancing, so whether it be in 5 or 10 years, CRISPR technology will be used in our daily lives. Now it is up to you to decide to accept it now or later.